Hello everybody, today we will be reading the paper Pupil Size Tracks Attentional Performance in Attention Deficit Hyperactivity Disorder by G. Wainstein, D. Rojas-Líbano, N. A. Crossley, X. Carrasco, F. Aboitiz, and T. Ossandón. Now this is a neuroscience paper, not machine learning, but we will be reading it the same exact way. Attention Deficit Hyperactivity Disorder, ADHD diagnosis, is based on reported symptoms, which carries the potential risk of over or underdiagnosis. A biological marker that helps to objectively define the disorder, providing information about its pathophysiology, is needed. A promising marker of cognitive states in humans is pupil size, which reflects the activity of an arousal network related to the norepinephrine system. We monitored pupil size from ADHD and controlled subjects during a visuospatial working memory task. A subgroup of ADHD children performed the task twice, with and without methylphenidate, a norepinephrine dopamine reuptake inhibitor. Off-medication patients showed a decreased pupil diameter during the task. This difference was no longer present when patients were on medication. Pupil size correlated with the subject's performance and reaction time variability. Two vastly studied indicator of attention. Performance. Furthermore, this effect was modulated by medication. Through pupil size, we provide evidence of an involvement of the noradrenergic system during an attentional task. Our results suggest that pupil size could serve as a biomarker in ADHD. Okay. So ADHD usually gets diagnosed by involvement with a doctor and talking to them. A doctor asks about symptoms and watches how a kid behaves and makes a call. But there is no blood test, no scan, or no number. And so there may be instances where they are misdiagnosed. So now what these researchers are doing are trying to prevent the potential risk of over or undiagnosis of ADHD symptoms. Sometimes kids get missed or sometimes they get overlabeled, and this is the problem that they are trying to fix. Now, off-medication patients showed a decreased pupil diameter during the task. This difference was no longer present when the patients were on medication. Now this result right here, off of their meds, ADHD kids had a smaller pupil response. Now once you put them on their meds, the gap basically disappears. And this is the finding of the paper, defended inside one single sentence. Now pupil size correlated with the subject's performance and reaction time variability. Now the pupil response also lined up with how well the kids did and how steady their reaction times were. So it tracks the actual behavior of the child as well. So now there are three claims being made here: the group difference, the medication normalizing it, and that pupils track behavior. All right. Attention Deficit Hyperactivity Disorder, ADHD, is the most prevalent childhood neuropsychiatric disorder. This condition is characterized by inattention, impulsiveness, and hyperactivity, and is becoming increasingly recognized that many patients continue having these types of difficulties in adulthood. Most ADHD symptoms are related to problems in behavioral and cognitive control and have been attributed to a deficient dopaminergic signaling. First-line treatment for ADHD is given by stimulants, mainly methylphenidate. And the usual second-line treatment is atomoxetine, a noradrenaline reuptake inhibitor. Both kinds of drugs increase the catecholamine availability at the synapses. This, together with evidence of a weak but consistent link between genetic polymorphisms associated with the catecholaminergic system and ADHD, prompted the widely accepted hypothesis that in this condition, there is an underlying deficit in catecholaminergic neurotransmission. Currently, diagnosis is being performed solely on the basis of observed behavior and reported symptoms, which carries the potential risk of overdiagnosis or underdiagnosis. Like any complex disorder, ADHD patients display clear heterogeneities at the clinical and biological level. These differences probably explain why some patients do not respond to any medication. A biological marker that helps to objectively define the disorder, providing information about its pathophysiology and potential responses to medication, is needed. So this last line is what they are looking for to prevent the over- and underdiagnosis of patients. Okay. A promising potential marker of cognitive states in humans is pupil size. After controlling for stimulus luminance, which means, so pupil size can also be determined based off of the amount of light inside the room. So what they're doing is they are controlling for luminance. So they are making sure that the light levels do not change the outcome of their research. Pupil size correlates with task difficulty, emotional valence, physical effort, motor output, and arousal states. Independent findings suggest that these fluctuations in pupil size reflect the state of the brain norepinephrine system. This system originates in the locus coeruleus, and projects throughout the cerebral cortex, hippocampus, thalamus, and midbrain, among others. Brain areas associated with attentional processing, for example, parietal cortex, pulvinar nucleus, superior colliculus, receive particularly dense LCNE innervations. Data from animal models, both rodents and non-human primates, have shown a central role for the LC in selective attention. So the first thing that they mention is your pupil doesn't only react to light. Once you control for the brightness, the pupil size tracks the mental effort and arousal required while doing a certain task. So it's basically a tiny effort meter in your eyeballs. Harder the task is, the bigger your pupils get. Now, deep inside of the brainstem, there is this little cluster called the locus coeruleus, or the LC. It's the brain's main source of norepinephrine, and it wires to almost everywhere in your entire body. The LC fires norepinephrine. Your pupils dilate as a response to that. Now, this norepinephrine system feeds straight into your attention circuits. So it follows this chain. The attention runs on norepinephrine. Norepinephrine comes from the LC, and the LC drives the dilation of the pupil. So the pupil reading it might tell you something about the attention of this person. This is the entire hypothesis stated inside this introduction. All right. Nowadays, pupil size can be monitored in a completely non-invasive way in humans using a remote camera and infrared light. We hypothesize that if pupil size reflects the activity of the LCNE system, which is one of the important attentional systems in humans, then pupil size is a potential and unexplored marker for attentional states in ADHD. If this is the case, changes in pupil size during an attentional task should reflect the behavioral differences observed between ADHD patients and control subjects. We monitored pupil size from ADHD and control subjects during a visuospatial working memory task. Deficits in these tasks are one of the most consistent impairments of executive functions in patients with ADHD. Instead, unaffected siblings present deficits in this type of task. Indeed, unaffected siblings present deficits in this type of task, which are intermediate between ADHD and controls, a finding that has led some authors to propose it as an endophenotype. In our task, 3 images plus a distractor were sequentially presented, and then a probe image was shown. Subjects had to evaluate if the probe image had been presented before. ADHD patients performed the task in 2 separate sessions on and off medication, which in all cases was methylphenidate. We found that pupil size changed during the probe presentation, the intentionally most relevant cue of each trial, and co-varied with the subject's variability in response time and performance. Furthermore, this effect was modulated by medication. Okay. So the if-then that they are trying to find is, if pupil size reflects activity of the LCNE system, then the pupil size is a potential marker for ADHD. So to experiment on this, what they did is they pick a memory task that ADHD kids are known to be bad at. But what they found out is that even the unaffected, healthy siblings of ADHD kids do perform a little worse on this task, which is why some researchers treat it as a heritable middle ground trait. Let's take a look at the results. As a first step, we performed the analysis using all the data, 28 ADHD subjects, 22 controls, and a subgroup of 17 of the 28 ADHD children who performed the task twice, on medication and off medication. Here we describe the data and compared between groups, both the behavioral and the physiological measures. As a second step, we specifically focused on the subset of subjects which performed the task on and off medication. In this way, we were able to assess for medication-associated changes on the same subject. These are the three groups that they performed this experiment on. 28 ADHD subjects, 22 controls, and a subgroup of 17 who performed the task twice. So ADHD is the full unmedicated group. Control is the non-ADHD kids, and MADHD are the 17 patients who came back and did it again on their meds. So MADHD isn't a different set of kids, it's the same ADHD kids, but on medication instead. Now let's take a look at this figure closely. What happens in the trial is basically the kid looks at a cross in the middle, then 3 dot arrays flash, one after the other. This is the cross, then array 1, array 2, array 3. Alright, then after that is a distractor image. Suppose a picture of an object or an animal, anything to kind of take the attention away. Then a probe, which is just 1 dot, and the question is, did this probe show up in one of the previous arrays? Yes or no? Now this probe moment is where they test whether the child is being able to pay attention, because that's where your brain has to fire all of their attention at once. Now this figure B is the performance by group. Each dot is one kid's accuracy and the bar is the group's average. Now control has the highest, ADHD has the lowest, and MADHD is somewhere in the middle. Now figure C has high load versus low load. Now load is just another word for difficulty. Low load means remember 1 dot per image, and high load means remembering 2 dots, so like 6 spots total. Everybody is worse on a higher load, which is good. It just means that their difficulty knob is actually working. Alright. Now task performance results. To assess for performance differences between groups, we first conducted a Crossley-Wallace analysis of variance, which gave a significant differences. Post-hoc test performed through Bonferroni method showed differences only between ADHD and control groups. We obtained the same differences via Tukey's honestly significant difference procedure. When trials were parsed by cognitive load, the data confirmed that 2 dot arrays were harder for these subjects across all groups. There were significant differences in performance levels for low and high cognitive loads as assessed by a Wilcoxon-signed rank test. The plot in figure 1C shows the change in performance for each subject related to low and high cognitive loads. Okay. Now Crossley-Wallace just asks, are these groups different at all? Without assuming the data forms a nice bell curve. Now they use these rank-based tests the whole paper because the sample is very small and slightly messy. Now the differences between ADHD and control groups here, ADHD versus MADHD. Now if you look at the p-values, only ADHD versus control is significant. The medicated kids aren't statistically different from the controls, but they're also not different from unmedicated. So on pure behavior, the medications nudge them towards normal. but it is a very soft effect. Now, here, Wilcoxon values for high versus low load. Alright, now this line just confirms that high load was harder for everybody. And that's all it says. Pupil diameter changes during the trial. Average pupil curves across trials in subjects reliably covaried with visual stimuli presentation in the 3 groups. Okay. Each curve in figure 2a, which corresponds to an average across subjects, shows diameter increased associated with the presentation of dot arrays, the distractor and the probe dot. On average, the largest increase in pupil diameter in the 3 groups occurred after a probe onset. To see individual pupil curves, see supplementary figure 1. To visualize and compare the actual maxima across trials, we performed a parallel analysis. Instead of looking at the average curve per subject, we used the same pupil time series to average the maximum pupil size for each subject in trial. After probe onset, these values are shown in figure 2b. To assess for difference between groups in maximum pupil size after probe onset, we conducted a Kruskal-Wallis analysis of variance, which gave significant differences. Post hoc tests conducted using the Bonferroni method showed significant differences between ADHD and MADHD, and also between ADHD and control, but not between MADHD and control groups. We obtained the same differences via Tukey's honestly significant difference procedure. As such, medication tended to normalize the pupil response of patients during the attentional task. Pupil diameter average magnitudes for the first 3 arrays presentations are shown in supplementary figure 2. Okay. So let's take a look at figure 2a. Now, time is given along the x-axis, and 0 is the probe. Now, up the side is pupil diameter. Each colored line is one group average, and watch that every little bump lines up with a dot array flashing, and then this huge spike right here. This is right after the probe. This is the pupils reacting to the mental work it takes to pay attention to this probe. Now let's highlight the difference between the red line and the blue line after the probe. So after the big spike, you'll notice that the control group sits the highest and the unmedicated ADHD group sits the lowest. And the medicated ADHD gets pulled back up towards the control group. So smaller pupil response when they're off their meds, that's the first claim, and that's visible inside this figure 2a. Now let's take a look at 2b. This is max pupil diameter after probe onset. So this is the same figure, but just in a dot plot instead. They grabbed each kid's peak pupil after the probe and they averaged it, and you can see that the control has the highest, ADHD the lowest, and MADHD somewhere in between. Now, it speaks on several differences between ADHD and MADHD, and also between ADHD and control, but not that much between MADHD and control groups. Now, when we check the p-values again, off meds versus controls, they're different. Off meds versus on meds are different, but the on medication group versus the control group, they are nearly identical. So this says that the medication didn't just help with their ADHD, but it also reset the pupil size back to normal. So this shows that the pupil is more sensitive than the behavior that it's supposed to be measuring. Associations between pupil diameter and behavioral variables. We then evaluated associations between pupil diameter and two of the most relevant behavioral variables, performance and variability in reaction time. In all the combinations, these variables showed consistent associations as assessed statistically by the Spearman correlation coefficient. We used the subjects from all three conditions to analyze these associations. The maximum pupil diameter measured after probe presentation was inversely associated to the variability in the subject's reaction time. On the other hand, maximum pupil diameter was directly associated with the subject's performance in the task, indicating that pupil size during the crucial part of the task was associated with the subject's outcome. Pupil size associated subject's outcome. Finally, consistent with a previous report and with two previous associations, we found that performance was inversely associated with variability in each reaction time. In terms of correlations within groups, we consistently found that the largest correlation values corresponded to the ADHD group, intermediate values to the MADHD group, and the lowest to the control group. Performance did not covary with raw reaction time. Furthermore, pupil diameter average magnitudes for the first three arrays presented positively covariates with array presentation in control and ADHD group. Nevertheless, this increase in pupil diameter does not correlate with performance in pupil diameter measured after probe presentation within subjects. Alright, so this third claim is, does the pupil actually track how well the kids did? Now for this, they pool everyone and use Spearman correlation, which just measures whether two things rise and fall together on a scale from -1 to +1. Now, here it says inversely associated with the variability in reaction time. Now, the bigger the pupil, the less jittery the reaction time, and that jitter is the inconsistency, which is a classic ADHD signature. Now, if you look at the value, Spearman correlation RHO minus 0.68, this is a solid negative correlation. Now, maximum pupil diameter was directly associated with the subject's performance in the task. So, row 0.63. This marker shows that bigger the pupil size, better the accuracy. Now, the largest correlation values correspond to the ADHD, intermediate MADHD, and lowest to the control group. So the correlation is strongest in the unmedicated kids and weakest in the control group. So the pupil behavior link is tightest exactly where the problem we are finding is. So the pupil behavior link is tightest exactly where the problem is with the patients with ADHD. Paid comparisons between conditions on and off medication. For a total of 17 subjects, we were able to collect one off medication session. Children suspended their medication 24 hours before performing the task, and a matching on medication session for the same subject. We use these data to evaluate differences between the on medication and off medication conditions for the experimental variables, both physiological, pupil diameter, and behavior. During on-medication sessions, children displayed a larger pupil diameter during probe presentation compared to off-medication sessions as assessed by a Wilcox-signed rank test. Performance, on the other hand, did not vary significantly between on and off-medication conditions. Neither did the standard deviation of the subject's reaction time. So in this, every kid is being compared to themselves. It's the same kid for 2 different days on and off medication. Children displayed a larger pupil size during probe presentation. These are the values. So within the same kids, the medication reliably boosted the pupil response. Performance, on the other hand, did not vary significantly between on and off conditions. Here. So this says that the accuracy didn't significantly change, and neither did their reaction time jitter. So the medications moved the pupil, but not statistically, the behavior in these 17 kids. So the marker is responding even when the behavior test cannot catch a change. Medication-associated changes in pupil diameter and behavioral variables. Finally, we analyzed the relations between pupil size and performance, and between pupil size and reaction time, specifically in the subset of subjects which perform the task in on and off medication conditions. To do this, we subtracted the mean variable values of the off medication sessions from the mean values of the on medication sessions, separately for each subject. We added the prefix delta to the new variables representing the on-medicated minus off-medicated values. We then plotted these differences against each other, see figure 5a. For the case of delta pupil diameter and delta performance, we found a positive association, as measured by Spearman correlation. In the case of delta pupil diameter and delta reaction time variability, we also find an association. These results indicate that if the medication was accompanied by a large increase in performance or a large decrease in reaction time variability for a given subject, then it was also accompanied by a large change in pupil diameter. Interestingly, we noted that the subjects who displayed a larger change in performance were subjects which performed the first task off medication. We've therefore next looked at mean values parsed by session irrespective of medication condition. We found that there was a small but significant effect of session on performance and not on pupil size. These results show that there could be an effect of medication on the observed pupil and reaction time changes, but in our data, there is a potential confound with training measured as session order. Alright, so for each kid, they take the change from off to on medications and they call it delta. Delta pupil, delta performance, delta reaction time. Then they ask, do the kids who changed most on one also change most on the other? So if we check here, rho is equal to 0.75, p-value, and rho is equal to minus 0.63. The kids whose accuracy jumped most on the medications also had the biggest pupil jump. And the bigger the pupil jump, the bigger the drop in reaction time jitter. So even though the average behavior change wasn't significant, the size of each kid's pupil change tracks the size of their behavior change. So this is an individual kid-by-kid link. Now let's take a look at this. The subjects who displayed a larger change in performance were subjects who performed the first task off medication. So this is the problem. The kids who improved most had done the off medication sessions first. So this improvement may also be because they just got better from practice, from having seen the task before, and not necessarily the drug at all. Small but significant effect of session on performance. This. So they tested and performance did improve from session 1 to session 2 no matter what, which is a practice effect. Performance did improve from session 1 to session 2 no matter what. Now this is a practice effect and it is real. Now this is a practice effect, but pupil size didn't show that session bump. But the pupil size didn't show that session bump. This could be an effect of medication on the observed pupil and reaction time changes. So they are being upfront on the fact that this boost in performance may just be because of the more amount of practice that children who did it twice were able to get. Discussion. We present evidence supporting pupil changes during an attentional task as promising biomarker for ADHD. It has been recently shown in animal experiments that pupil diameter tracks arousal and attention states. Specifically, pupil diameter co-varies with neuronal activity in the LC-NE system, so-called arousal or alerting network. The LC-driven norepinephrine modulation, which reaches almost the entire cortical surface, has a direct readout in pupil size. This confluence of evidence from any involvement in attentional circuits and control of pupil size prompted us to explore pupil size as a biological marker in ADHD. We tested ADHD and controlled children in visuospatial working memory task, previously known to be challenging for ADHD patients. A subset of ADHD children performed the task both on medication and off medication conditions. Off medication ADHD patients showed decreased pupil diameter response during the task compared to controls. On average, this difference was no longer present when patients were on medication. Specifically, we saw a task and event-related pupil response, which tracks the array presentations and displays the largest increase during the probe dot presentation. When the probe dot was presented, subjects had to pay attention to it, compare it with what they recall about the previous 3 arrays, and make a decision. In this crucial moment of the tasks, the size of the induced pupil response helped to distinguish between patients and control subjects. Given that pupil diameter reflects the activity of the LC-NE system, these results supported the involvement of the cerebral NE system in ADHD. We found a correlation between pupil size and subjects' performance, measured as a response accuracy and reaction time variability in the ADHD group. These performance metrics are 2 vastly studied behavioral ADHD markers in attentional tasks. In this study, the correlation between these markers and pupil size were weaker in the on-medication group and displayed their lowest values in the control group. This association between pupil size and behavior seems to be restricted to the period after probe onset. For example, the sustained baseline increase across array presentations did not correlate with response accuracy or reaction time variability. It has been proposed that these manifestations of ADHD, increased reaction time variability and poor accuracy in attentional tasks, could be caused by transient disruptions of attentional neuron network activity. These disruptions are in turn caused by an increase in the activity of other internal networks, such as the default mode network. In addition, recent models have proposed the role of LC-NE circuits in so-called mind-wandering periods, which might potentiate the default mode network and decrease the activity of attentional networks. This interference is characterized by a decrease in the phasic gain of the LC-NE system, which in turn causes decreased pupil diameters. Thus, there are compelling reasons for using pupil size dynamics to characterize and diagnose ADHD. We also found that the subjects whose performance and reaction time variability changed more with the medication also displayed larger pupil change between on-medicated and off-medicated conditions. All right. Hence, the covariation between pupil size change and behavioral change seems subject-specific. We note that the session order, on medicated or off medication, was randomized in our subjects, which means that some of them learned the task off medication and they were tested a second time on medication. Besides the performance, pupil diameter and reaction time variability did not consistently change across trial box. This suggested that motivational or learning effects were not modulating these responses. Overall, the association between pupil diameter changes and ADHD behavioral performance markers and how these associations differ between ADHD and normal children suggests that pupil diameter changes during a visual spatial working memory task may be a useful biomarker of ADHD. These associations are also consistent with the hypothesis that ADHD is a neurodevelopmental disorder related with the broadly distributed NE system, and not only a problem confined to a prefrontal striatal dopamine circuit, as implied by the classical version of the dopaminergic hypothesis of ADHD. Okay. So this is just summarizing everything that we have already read about. This off-medicated ADHD showed decreased pupil diameter response. They lean on this again. The strongest results are in the children with ADHD who are off medication. Now, these manifestations of ADHD could be caused by transient disruptions of attentional network activity. Now, the theory that they are trying to plug in is that the default mode network is your daydreaming, mind-wandering network. And the idea is in ADHD, it barges in when it's supposed to stay quiet. And so these intrusions, they show up as lapses or jittery reaction times, and they argue that this dips in the pupil response. Now, they flag it as a theory that they're borrowing and not something that is proved in this paper. Now, here. These associations are also consistent with the hypothesis that ADHD is a neurodevelopmental disorder and not a problem confined to a prefrontal striatal dopamine circuit. Now, the old story was that ADHD equals a dopamine problem in one little frontal circuit. Now, the pupil data points at norepinephrine instead. So, this system blankets the whole brain, and so they're nudging this field of research from a local dopamine point towards norepinephrine, which goes everywhere. So their big claim is that ADHD is NE distributed and not just dopamine local. Now let's take a look at methods. Methods. Subjects. A total of 50 subjects participate in the study. 28 subjects are patients diagnosed with ADHD. All ADHD children were being treated with methylphenidate. 22 subjects were non-ADHD assigned to our control group. A subgroup of 17 ADHD patients performed the task twice, on and off medication. The order of these sessions, on or off medication, was randomized to avoid carryover effects. In the on-medicated session, ADHD subjects took their regularly prescribed dose 2 to 2.5 hours before the task, which is the time corresponding to the maximum drug concentration after the oral administration. For off-medication sessions, children discontinued their medication 24 hours prior to the day of testing, which is enough time to remove 80% of the circulating drug levels. No significant differences were found between the groups in age, IQ, or educational level. The ADHD group had a mean IQ of 100, while the non-ADHD group had a mean IQ of 105. All ADHD subjects were diagnosed as ADHD combined subtype by a trained child neurologist according to the DSM-4 criteria. All participants were also screened for neurologic or psychiatric comorbid conditions using a protocol that included parents' interview, MINI kid test, and general psychological and physical assessment of the children. The Connors abbreviated parent-teacher questionnaire is usually utilized to screen for symptoms of ADHD in the clinical setting. In the present study, it was used as an additional symptoms counting tool. Participants with ADHD and those from the non-ADHD group were evaluated using this instrument as a controlling variable. Parents granted informed consent of their child's participation, and children signed an informed assent form. The procedures in the study were approved by the Ethics Committee of the School of Medicine of the Pontificia Universidad Católica de Chile, in accordance with the Declaration of Helsinki. Okay, so there were 50 subjects, 17 with ADHD, 22 with non-ADHD in the control group. All right. Now, this is a very small sample size, and the 2 sessions for the MADHD group were about 6 months apart on average, with a huge variation. And a lot of changes in a kid happen over 6 months. Now, this is not very fatal, but they say it because it caps how strong any single result can be. Now, they took their regularly prescribed dose 2 to 2.5 hours before the task, and they discontinued their medication 24 hours prior. Now, this is enough to remove 80% of the circulating drug levels. Now, the medication setup On the on day, they time the dose so the drugs like peaks at the right time. And then on the off day, it's 24 hours of waiting so that the medication can clear out of their body. Now, every patient here is a combined subtype of inattentive plus hyperactive. So this might not carry over to the purely inattentive kids, but they are all just kind of subcategorized into ADHD uh, diagnoses. Subjects performed a Sternberg type delay visuospatial working memory task adopted from Dolkos and McCarthy. Memoranda were 1 or 2 dot arrays with the dots located variably in any of the 16 spaces of a 4x4 grid. On each trial, subjects were instructed to start by fixating on a black cross located at the center of the screen. 500 ms dot array presented commenced. 3 different dot arrays were presented on each trial. Each array was presented for 750 milliseconds with a 500 millisecond delay between arrays, during which a fixation cross was presented. After the last delay period, a distractor image was presented, then a probe dot was presented for 1.5 seconds. This is a dot within the grid, and subjects had to answer yes if it had been present in one of the previous arrays, and no if not. Immediately after probe offset, we provided a feedback image for 1,500 milliseconds. Participants were instructed to respond as fast as possible. 3 distractors, a task-related dot array, neutral natural image, emotional image. They were constructed and modified from public domain images adapted from Vidal, Ossandón et al. In 25% of the trials, no distractors were present. Trial types were presented randomly. There were also 2 trial types according to the cognitive load. In low load trials, only 1 dot was present. In high load trials, 2 dots were present. Now, this is what we described while looking at this previous diagram here in figure A. This was the test shown, and these are the arrays of different dots, the distractor image and then the probe image. All right. Data acquisition. Pupil diameter data was acquired with iLink 1000 with a 1 kHz sampling frequency. Subjects sat in front of a table containing the computer screen for image presentation and the eye tracker device. During the task, subjects kept their head in a forehead/chin rest. Subjects were placed in a viewing distance of 30 centimeters. Illuminance data with lux meter positioned in the same chin rest used by the subjects at the same distance from the screen. Only modestly large difference in illumination occurred between delay and array displays. The distractor presented more variability in illuminance and had lower illuminance values. All right, behavioral data analysis. Stimuli were presented using presentation software, and subjects delivered their responses using a keypad. Alongside checking for correct responses, we also recorded the reaction time as the time delay between the probe presentation and the subject's response. This allowed us to calculate the variability in reaction time, a parameter known to correlate with the cognitive performance in ADHD children. Pupil data analysis. Pupil diameter analysis was performed using MATLAB, software with in-house functions. First of all, period of blinks, in which no pupil diameter information was available, were detected by the iLink software. Pupil data surrounding blinks were removed from the time series using the analysis. Pupil diameter during these periods were estimated using a cubic spline interpolation, implemented through MATLAB function spline. To obtain the pupil diameter average profile, data of each participant were baseline adjusted and smoothed by a bandpass bandwidth filter. A low-pass frequency filter, a pass frequency filter applied to detrend the basal slope change. Unless otherwise stated, data analysis was restricted to 8-second trial. After filtering and outlier removal, the pupil time series was normalized by means of a Z score separated for each trial. To calculate the pupil diameter maxima during probe presentation, we use the same Z score and pupil time series is superior to it. We find a window between 500 milliseconds after probe onset and 500 milliseconds after probe offset. Within that window, we calculate the maximum value of pupil diameter for each trial. We then computed the mean value of this maxima separately per subject to obtain data in figure 2. So they took thousands of samples of a second eye tracker. Every time they blink, it leaves a gap. So they patch the gaps by interpolating, and then they filter out the super fast noise and the super slow drift to isolate the actual response. Then there's a Z-score for everything, so the kids are comparable. Now, statistics. All our statistical tests were implemented in MATLAB. Due to the non-normal nature of the data, we used non-parametric tests to assess statistical differences. For paired comparisons, we used the Wilcoxon sine rank test with MATLAB function sine rank. For non-paired comparisons, we implemented permutation tests using a t-statistic. Then implemented the permutations and calculated the t-statistic for each permutation.
To assess significance, we compared the non-permitted t-value with the distribution of permitted t-values and calculated if it lied within the 95% of the distribution. All tests were performed using the subject and the unit of analysis. Data availability, authors declare that the data gathered are available within the paper. They also state that data are all available from the authors upon reasonable request. All right, now this is the end of this paper. All right, so thank you so much. This is the end of this paper. Thank you so much for reading this with me. Thank you.